All right, today we're jumping into something really important. We're going to look at how a single fundamental flaw in our genetic map could be leading to misdiagnoses and, more importantly, how scientists are now fixing it. So, let's just start with a big question. What if the very map our doctors and scientists use to navigate our DNA has these huge, blurry, incomplete sections? And what if just filling in those details could completely change a diagnosis for someone? To really get what's going on, we first need to talk about the tool that basically everyone in this field uses. Think of it as our genetic encyclopedia.
That tool is called gnomAD, and it's exactly what it sounds like, a massive, massive database. It's pretty much the most comprehensive catalog of human genetic differences ever put together, built from the DNA of hundreds of thousands of people from all over the world.
Okay, so here's the crucial part. How do doctors actually use it? Well, they look at something called allele frequency, and that's just a fancy way of asking how common is this specific genetic typo. You know, if it shows up all the time in the population, it's probably harmless. But if it's super rare, that's a red flag. It might just be the cause of a disease. Getting this right is absolutely critical. But here's where that map we talked about starts to get really blurry. It turns out the way gnomAD groups people can sometimes hide the real story that's written in their DNA. The problem is that the labels are just too broad. For instance, someone might get put in the Admixed American or African/African American category. Now, that's useful to a point, but these are huge, incredibly diverse groups of people. The reality is that so many of us have this beautiful, complex mosaic of ancestry from different continents, and that crucial detail just gets lost in these big buckets. In this image right here just perfectly shows the problem. This is a genetic map that plots people based on their overall or global ancestry. Now look closely. On this big picture map, individuals B and C look almost identical. I mean, they're clustered in the exact same spot, which would suggest they have pretty much the same genetic background.
But now, let's zoom in. This is what's so cool. When we actually look at their chromosomes, piece by piece, the story completely changes. The different colors you see represent ancestry from different continents, and boom, suddenly B and C look nothing alike. Their internal genetic mosaic is totally unique. That's the hidden detail the big map misses entirely.
So the big question is, how do we get this high-resolution view for everybody? Well, the answer is a technique that's a bit like painting the genome. This technique is called local ancestry inference, or LAI for short. And instead of just giving a person one big overall ancestry label, LAI goes through their DNA segment by tiny segment and paints each piece with its specific origin. It's like switching from a blurry photograph to a 4K ultra-high-def scan. I love this analogy. Think of it this way. Global ancestry is like knowing a beautiful mosaic is made of Italian glass. Okay, that's interesting, but it's not very helpful. Local ancestry, on the other hand, is like having the full blueprint that shows you exactly where every single red, blue, and green tile is placed. You finally see the real, detailed picture. This schematic kind of pulls back the curtain on how it works. You can see how LAI paints the chromosomes of these three individuals with different ancestral colors. Now this reveals something amazing. A specific variant might only show up on a chunk of chromosome from one specific ancestry. And that is the game changer, because it means we can now calculate a much more accurate frequency for that variant, but within that specific ancestral group. So what does this new high-res picture actually mean in the real world? Well, it's not just a small tweak. We're talking about a genuine revolution in how clinical genetics is done. Get ready for this number. In the African/African American group alone, a staggering 85% of genetic variants show at least a 2 times difference in how common they are when you compare the African ancestry segments to the European ancestry segments. This isn't a small correction. It's a massive shift in our understanding. And here's a perfect real-world example. On the left, you see the old, blurry picture. A variant linked to type 2 diabetes risk looks like it has a frequency of about 24% in the broad Admixed American group. Okay, but now look to the right. Using this high-res technique, we see the real story. That same variant is actually present in 45% of the DNA segments that come from Amerindigenous origins. The risk is way more concentrated than we ever knew. Or take this case, a variant linked to a much higher risk of kidney disease. If you look across the entire GenomeAD database, it seems pretty rare, only about 1%. But LAI shows us that within the African ancestry DNA segments of individuals in the African/African American group, the frequency is actually 27%. Imagine you're a doctor, making a decision based on a 1% risk versus a 27% risk. That changes absolutely everything.
And this chart here, it shows a really clear pattern. This isn't random. When these variant frequencies are different, they are overwhelmingly more common in the African ancestry DNA segments. We're talking 69% of the time. This just highlights exactly where the most important information has been hidden all along. So what's the bottom line? What's the real payoff for patients? It's this. One of the absolute worst things to hear from your doctor is that you have a variant of uncertain significance. It's terrifying. It means they don't know if it's harmless or harmful, and it can lead to years of anxiety and endless tests. By showing that a variant is actually way more common within a specific ancestry, this new method can help reclassify it as benign, providing clarity, relief, and peace of mind.
Okay, so let's bring this all together. What does this all really mean for the future of medicine and, well, for all of us?
See, this isn't just about better data. It's about better care. It means we get much more accurate genetic risk assessments, especially for people with mixed ancestry. It means fewer of those dreaded, uncertain diagnoses. And of course, that directly leads to less patient anxiety and helps avoid unnecessary, sometimes painful, follow-up procedures. At its core, it's about building a smarter, fairer, and more precise genetic map for every single person.
And don't think for a second this is some rare niche thing. Get this. Over 81% of all the variants they analyzed got a new, higher maximum frequency once local ancestry was factored in. That means for more than 4 out of every 5 variants, our understanding of how common it is, and therefore how risky it might be, has just been massively improved.
What this work really reminds us is that our DNA isn't some single, flat map. It's a rich, multi-layered tapestry woven from the incredible stories of all of our ancestors. And that leaves us with a really important question to think about. As we continue to map this tapestry with ever greater precision, just how profoundly will it reshape the future of medicine for all of us?